Hello students, welcome to chapter 10. We will discuss photosynthesis. And firstly, photosynthesis is going to occur in an organelle called the chloroplast. And photosynthesis is going to stand for photo, meaning light. And synthesis means we will produce glucose. So in photosynthesis, this is the process of converting solar energy into chemical energy in the form of glucose within the chloroplast, which is located in plants and other photosynthetic organisms. Directly or indirectly, photosynthesis nourishes almost the entire living world. Autotrophs are self-feeders. Auto means self, and photoautotrophs are self-feeders that use sunlight in order to produce their own food. And the food is going to be glucose. There's other types of autotrophs. Chemoautotrophs do not use the sunlight. However, they use other chemicals, such as chemicals from the soil, in order to synthesize their own food. Photosynthesis occurs in plants, algae, certain unicellular eukaryotes, and some prokaryotes. So photosynthesis occurs in photosynthetic organisms. We can see here two beetles, or organisms here, that are benefiting from photosynthesis as they consume this leaf. Again, here are the different examples of photoautotrophs: plants, multicellular algae, unicellular eukaryotes, purple sulfur bacteria, and cyanobacteria are going to be photoautotrophs. Heterotrophs obtain their organic material from other organisms. So hetero is the opposite of auto. Heterotrophs are going to be mostly consumers because they eat other organisms which are going to provide the organic material for the organism and also the energy needed. Almost all heterotrophs, including humans, depend on photoautotrophs for food and oxygen. Fossil fuels were formed from the remains of organisms that died hundreds of millions of years ago. When we look at the different strata or rock layers from Earth, we can see different fossils that are embedded in those rock layers. These fossil fuels are going to be rich in carbon-hydrogen bonds, being hydrocarbons, and this is what makes them so energy rich. Gasoline is an example of fossil fuels that we use in order to power many different motors, such as your gas-powered cars. Chloroplasts are structurally similar to and likely evolved from photosynthetic bacteria. Chloroplasts having their own genetic material, having their own DNA. The structural organization of these organelles allows for the chemical reactions of photosynthesis. If we take a look at the anatomy of a plant, the plant has leaves, and within the leaves we have a layer called the mesophyll layer. This layer of tissue in the leaf is going to have these cells called mesophyll cells. And mesophyll cells are going to be the cells where most of the photosynthesis is going to occur. So mesophyll cells are going to be the site of photosynthesis because they have the most chloroplasts. Stomata are little pores or holes under the leaf, which allow CO2 to enter the plant and oxygen to exit the plant. This allows for plant respiration through the pores or holes called the stomata. Let's take a look at the anatomy of a chloroplast. So the chloroplast mainly has two areas. So we have the stroma, which is the fluid that fills the interior of the chloroplast, and we have the thylakoids, which are these uh, membrane sacs. They kind of look like coins that are going to be the third membrane system of the chloroplast. If we stack several of these thylakoids or coins, we get a grana, which is just a stack of thylakoids. Chlorophyll is a pigment which absorbs light. Chlorophyll is going to be green in color because it reflects green wavelength of light. Chlorophyll is located in the thylakoid membranes and not the stroma. And now we have a picture of a leaf, and if we zoom in on the leaf, we can see that the leaf has a layer called the mesophyll layer, which is going to contain the mesophyll cells. And if we look at a single mesophyll cell, it has several chloroplasts in it. And if we zoom in on the chloroplast, we can see that the chloroplast has an outer membrane right here and an inner membrane here. And we have the innermost membrane, which is the thylakoid. So the innermost membrane, or the third membrane system, is the thylakoid. And the thylakoid is going to be each of these coin-shaped discs right here. So what happens when you stack those thylakoids together? Well, that is going to be called a granum. A stack of thylakoids is called a granum. And also, when we go back to the picture to the right, we see the stomata, which is going to be an entry and exit way for CO2 and oxygen. CO2 is going to enter the leaf, and oxygen is going to exit the leaf. Here's the chemical equation for photosynthesis, which is the opposite of cellular respiration. The inputs are going to be CO2, water, and solar energy. So these are your reactants. And your outputs are going to be glucose, oxygen, and water. Okay, so remember that these are your outputs, and also right here we have glucose. Chloroplasts are going to split the water into hydrogen and oxygen. This is how we're going to get oxygen as a byproduct of photosynthesis. Incorporating the electrons of hydrogen into sugar molecules. So we get the hydrogens from splitting the water, and that's how we're going to get the hydrogens for glucose, and releasing oxygen as a byproduct. Good. So right here we see where all of the hydrogens come from. If we follow the splitting of water, we split water into oxygen, and we split water into the hydrogens, and the hydrogen is going to be used to create water again, and also the 12 hydrogens and glucose are going to come from the splitting of water. So where does the carbon come from for the glucose? It comes from carbon dioxide. And the oxygen from glucose comes from CO2, and also the water comes from CO2. Photosynthesis is also going to include redox reactions. So we have the reduction here of CO2 into glucose. So CO2 becomes glucose because we are going to gain hydrogen ions and its partner electrons. And for H2O, we're going to become oxygen. So H2O becomes oxygen. So we lose hydrogens to get oxygen and also the electrons. So this is called oxidation. So in this case, CO2 was reduced and H2O was oxidized. Okay. Photosynthesis instead of cellular respiration is going to be considered a endergonic process. Instead of releasing energy, energy is going to be absorbed in order to synthesize a more complex molecule. And in this case, that molecule is glucose. Photosynthesis is going to have two steps. Photosynthesis has two steps. The first step of photosynthesis is called the light reactions, and the second step is called the Calvin cycle. Both of these parts of photosynthesis, remember, are going to occur in the chloroplast. And the chloroplast is going to have two parts, the thylakoids and the stroma. Step number one, the light reactions is going to occur in the thylakoids of the chloroplast. So what happens in the light reactions? In the light reactions, water is going to split into, like we saw earlier, oxygen and hydrogen. Oxygen, therefore, is going to be released as a byproduct. NADP plus is going to be reduced into NADPH. And we are going to phosphorylate ADP into ATP. This process is called photophosphorylation. So here we see a chloroplast. So this whole thing is the chloroplast. And the chloroplast is going to have two main areas: the stroma, which is the fluid that fills the chloroplast, and the thylakoid, the third membrane system, or the innermost membrane of the chloroplast. Okay, and then we have our first part of photosynthesis, the light reactions, which occur in the thylakoid, and the second part of photosynthesis, the Calvin cycle, occurs in the stroma. Notice how the inputs are going to be located at the top. So here's the inputs for photosynthesis. But as you can see, the inputs are going to enter either in the light reactions or the Calvin cycle. They don't all go to one part of photosynthesis. So light and water go into the light reactions, and CO2 goes into the Calvin cycle. And on the bottom right here, we have the outputs. So oxygen and glucose, the sugars. And so the output for the light reactions is oxygen, but for the Calvin cycle, this is where glucose is going to be synthesized as a output or products. So remember, input is going to be your reactants, and your output is going to be your products. Okay. So again, chloroplasts are the organelles where photosynthesis is going to happen. And chloroplasts are solar powered. So they require sunlight or solar energy in order to go through photosynthesis. Their thylakoids are going to transform light energy into chemical energy in the form of ATP and NADPH. Okay. Next, we're going to go over what light is. So light is a form of radiation, electromagnetic radiation. It's a form of energy, electromagnetic energy. And light is going to have different wavelengths. So light is a form of energy or radiation that has different wavelengths. So most of the wavelengths are going to be invisible to the human eye. The range of wavelengths is called the electromagnetic spectrum, which shows you the wavelengths of the radiation. The radiation that we are able to see with the human eye is called visible light, which is a very small part of electromagnetic spectrum. So this is going to range from 380 nanometers to 750 nanometers. And visible light is going to include light that is going to be used in photosynthesis. So visible light is absorbed during photosynthesis. Packets or particles of light are called photons. So here's the electromagnetic spectrum. So most of these different wavelengths or types of radiation, so gamma rays, X-rays, UV rays, infrared, microwaves, and radio waves, we cannot see with the human eye. So some other species of animals can see, but we cannot see it. So if we go from the left to the right, this is going to be go from a shorter wavelength all the way to a longer wavelength here. Okay, and we can see only a small sliver of the spectrum is considered visible light. So this is the only visible part. And if we kind of expand that, we're going to go from 380 to 750 nanometers. And if you see right here, each wavelength is going to correspond to a certain color. So if we're looking at 450 to 500 nanometers, this is going to correspond to blue light, blue wavelength. So, and what about green light? Green light is going to have a range of 500 to 550 or 570 nanometers. So each color of light has its own respective wavelength. Lastly, if you look right here, again we see as we go from left to right, shorter to longer wavelength, the shorter wavelength is going to have higher energy, whereas the longer wavelength you have means you have a lower energy. So what are pigments? So pigments are substances that absorb visible light. And an example, or the most predominant pigment in plants, are called chlorophyll. And chlorophyll is green in color. So why is chlorophyll green? Chlorophyll is green because it's going to reflect green light. So chlorophyll is unable to absorb green light. And earlier we saw that green light was about 500 nanometers to 570 nanometers. So this range of light, chlorophyll is not able to absorb. But the other ranges of light, the other colors, chlorophyll is able to absorb. So this is why we're able to see chlorophyll as green, and plants look green. It's because that reflected light is going to enter our eyes. Okay, so I just drew an eye right here. Not the best eye, so your eye right here. So the green light that reflects off of the chlorophyll is going to enter our eyes, and we're going to process that green light as green. And if you look at the other wavelengths of light, the other colors, those are going to be absorbed and not reflected. So here we see a video going over the pigments.
The thylakoid membrane in chloroplasts contains light-absorbing molecules called pigments. Different pigments absorb light of different wavelengths. One pigment, chlorophyll A, absorbs mainly blue-violet and red light. This molecule participates directly in the light reactions. Another molecule, chlorophyll B, absorbs mainly blue and orange light. We do not see these absorbed colors when we look at a leaf. Instead, we see the green wavelengths that are reflected back from the leaf or transmitted through it.
So again, in this video, we see that green light is reflected, but the other colors or wavelengths of light are absorbed instead. And that's why we're able to see green, or plants are green to us. Okay. A spectrophotometer is used to measure a pigment's ability to absorb various wavelengths. This machine is going to send the light through pigments, your sample, and measures the fraction of light that is transmitted at each wavelength. So there are two ways to express the degree to which a pigment absorbs a particular wavelength of light. So you can either absorb the transmittance or the absorption. So absorption is a measure of the amount of light that is absorbed by the pigment, and transmittance is the amount that's not absorbed by the pigment. And absorption spectrum is a graph plotting a pigment's light absorption versus the wavelength that we're looking at, the color of light. Okay, so here is a slide going over the spectrophotometer, and it is going to show us the transmittance of a certain wavelength of light. And so we start with our light source here, which goes through a refracting prism, separating the visible light into different colors or different wavelengths. And we are able to adjust the spectrophotometer to only let one wavelength of light go through. So in this case, we picked green light, which was 500 to 570 nanometers. And the wavelength that we picked is going to go through a sample. So in this case, if you look at green light, it looks like it goes completely through the sample. So if that light goes through the sample, it's going to hit a sensor, the photoelectric tube, and show us how much of the light is going to be transmitted through the sample. So in this case, it's about 100% transmittance. So 100% transmittance is going to be like saying 0% absorption. Okay. So 100% transmittance means low or 0% absorption. So our sample, which is chlorophyll, is going to absorb 0% of that green light. On the other hand, if we, our example 2, okay, we have the light again, the light source, and this time we're only letting blue light go through. And as you can see here, some of the blue light was absorbed by the chlorophyll sample. So again, we measure the transmittance of the blue light with the sensor, and in the meter we see that it's a little over 0%. So we have a very low transmittance, which indicates a high absorption, as we can see right here. So it's not 100% absorption, but it's close to it in this case. So our sample chlorophyll is able to absorb blue light very well. There are 3 types of pigments in chloroplasts. We have chlorophyll A, chlorophyll B, and carotenoids. Chlorophyll A is the main key light capturing pigment. Chlorophyll B is an accessory pigment, and also carotenoids are accessory pigments. So why do we have these extra pigments? Well, they're going to increase the plant's capacity to absorb different wavelengths of light for photosynthesis. The absorption spectrum of chlorophyll A suggests that violet blue and red light works best for photosynthesis. So for chlorophyll A, it absorbs violet blue and red light the best. And since chlorophyll A is green, it's going to not be able to absorb green light, but it reflects it. An action spectrum, it profiles the relative effectiveness of different wavelengths of radiation in driving a process. So in this slide right here, we can see our absorption spectra right here for graph A, our action spectrum right here for graph B. So in graph A right here, we have 3 pigments, chlorophyll A, chlorophyll B, and the carotenoids. And we can see in which wavelength do they have optimal absorption. So all we need to do is look at the peaks right here. So for chlorophyll B, the optimal absorption is going to be around between like 450 and 500. But if we look at the green range right here, 500 to like 570, we can see how none of these pigments really are going to have high absorption. They're going to, so if they have a low absorption in this range right here, it means that they have high reflection. So remember, students, low absorption is basically saying high reflection. Okay. So that explains why chlorophyll B and chlorophyll A are not green. Or I'm sorry, it explains why chlorophyll A and chlorophyll B are green and not the other colors right here, like purple or red. Also, if you look at the action spectrum right here, it shows you on the y-axis the rate of photosynthesis. So the effectiveness of photosynthesis at different wavelengths of light. So we can see that light is going to be absorbed the best from 400 to 500, around 425, and at a little less than 700, like 680. So around 425 nanometers, which is like purplish light, and 680, which is about like orangish light. The action spectrum of photosynthesis was first demonstrated in 1883 by Theodore W. Engelmann. In his experiment, he exposed different segments of a filamentous algae to different wavelengths, and areas receiving wavelengths favorable to photosynthesis produced excess oxygen, which is a byproduct of successful photosynthesis. He used the growth of aerobic bacteria clustered along the algae as a measure of oxygen production. The action spectrum for photosynthesis is broader than the absorption spectrum of chlorophyll. Accessory pigments such as chlorophyll B broaden the spectrum used for photosynthesis. So the main pigment is going to be chlorophyll A, but the presence of chlorophyll B and the carotenoids are going to provide us with a broader spectrum for absorption of different wavelengths of light. So that's the purpose of the accessory pigments. The difference in the absorption spectrum between chlorophyll A and B is due to a slight structural difference between the pigment molecules. So here we see the difference, which is only that functional group right here. So we have CH3 in chlorophyll A versus CHO in chlorophyll B. Accessory pigments called carotenoids may broaden the spectrum of colors that drive photosynthesis. Some carotenoids function in photoprotection. They absorb excessive light that would damage chlorophyll or react with oxygen. So not only do accessory pigments provide a broader range of light absorption, also it's going to protect chlorophyll from excessive light. So when a pigment absorbs light, it goes from a ground state to an excited state, which is unstable. When excited electrons fall back to the ground state, excess energy is released as heat. In isolation, some pigments also emit light in afterglow called fluorescence. Okay, so when pigments absorb light, there's going to be electrons in the pigment that are going to become excited. So when they become excited, we say that those electrons are in an excited state. And as we go from an excited state back to the ground state, energy is going to be released as heat, or energy can be released as light. And in some organisms, that emitted light is called fluorescence. So here we see an example of fluorescence as the excited state of the electron from the pigment returns to the ground state. So from 1 to 2, when we go there, heat or light is going to be released as energy. Now we're going to take a look at the structures of the thylakoid, where the light reactions are going to happen. the first part of photosynthesis. So before we do, let's go over the photosystem. So the photosystem is going to be the complex that receives the light initially. So the photosystem is going to be composed of a reaction center and light harvesting complexes. So the light harvesting complexes are going to be the outer part, the reaction center more of the inner part. And the reaction center complex is an association of proteins holding a special pair of chlorophyll A molecules and a primary electron acceptor. So the reaction center complex is part of the reaction center. Okay, so again, when we look at part of the photosystem, we have the light harvesting complex. This consists of several pigments that are going to pass along the photon of light to the reaction center. The light harvesting complexes transfer the energy of photons, again, to the chlorophyll A molecule in the reaction center. So these chlorophyll A molecules are special because they can transfer an excited electron to a different molecule, which in this case is going to be the primary electron acceptor. And so we are going to talk about the primary electron acceptor, which is going to accept the excited electron from chlorophyll A in the reaction center. The solar power transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of the light reactions. And so it's better to look at this diagram and we will understand the terms we went through. So the first step is for the photon, which was the particle of light, to hit the light harvesting complex of the photosystem. So in this picture, we see a photosystem. So there's two photosystems, photosystem 1 and 2. And this photosystem right here has two parts. So the outer purple part, the dark purple part, is called the light harvesting complex. And the light harvesting complex is also going to have several of these pigments, these chlorophylls, that are going to transfer the photon and pass it along eventually to the middle of the photosystem, which is the reaction center. The reaction center is going to have the chlorophyll A molecule in the bottom. And it looks like a pair right here. And also we have the primary electron acceptor. So the photon is going to first hit the light harvesting complex, and it's going to be passed along these pigments, and it's going to reach the reaction center by being passed along to chlorophyll A right here. So in chlorophyll A, there's going to be an electron that is now going to be excited by the photon that was passed to it. And that electron now goes from the ground state to the excited state, and that excited electron is transferred to the primary electron acceptor. And next, that excited electron can start to be transported along the electron transport chain in the thylakoid membrane. Okay. So again, there are two types of photosystems in the thylakoid membrane. We have photosystem 2 and photosystem 1. So the numbering in the photosystems is a little bit counterintuitive because photosystem 2 is going to come first in line. The reason why photosystem 2 is called photosystem 2 is because this is going to reflect the order of discovery. So photosystem 2 was discovered after photosystem 1. That's why it's called photosystem 2. But photosystem 2 is going to be first in line in the electron transport chain. The reaction center, chlorophyll A of photosystem 2, is called P680 because it's best at absorbing wavelengths of 680 nanometers. And we saw earlier in our range of wavelengths, 680 is kind of orangish to reddish light. Photosystem 1 is best at absorbing wavelengths of 700 nanometers, and that is why chlorophyll A in photosystem 1 is called P700, because chlorophyll A is best at absorbing P700 or 700 nanometers of light, which is around, it's like a red color of light. During the light reactions, there are two possible routes for electron flow, cyclic and linear. Linear electron flow is the primary pathway. This is going to involve both photosystems and produces ATP and NADPH using light energy. So remember, students, the light reactions is just part one of photosynthesis. So let's go through the 8 steps of linear electron flow. So our first destination is photosystem 2. The photon of light is going to hit the pigment that's located in the light harvesting complex of photosystem 2. The photon is going to be passed along these pigments, and eventually it's going to reach chlorophyll A, which is P680, so chlorophyll A. Okay. An excited electron from P680 is transferred to the primary electron acceptor. And now we call the primary electron acceptor P680+. And remember, students, the P680 and the primary electron acceptor are going to be located in the reaction complex. Okay, so that's important to know. So P680 and the primary electron acceptor are part of the reaction complex of photosystem 2, and that's different from the light harvesting complex. Okay. So again, here we see the location of the light reactions, which is located in the thylakoid. We have light come in, but also we have water come in, and our byproduct is going to be oxygen and NADPH and ATP. So here's the 8 steps represented on a diagram. So we have, again, light is going to first hit the pigment here in the light harvesting complex in photosystem 2. So this is PS2, and PS1 is right after, second in line. So the photon of light is going to be pass along the pigments and eventually reach chlorophyll A, and called P680, and it's going to the photon is going to hit a electron in P680. Where did the electrons come from? So if you look right here, we have water that's going to be split into oxygen and hydrogen. And remember, if we split water into hydrogen, hydrogen's partners are going to be the electrons right here. So the light helped split the water into its separate components. The electrons travel to P680, and the photon, when it hits the electron in P680, is going to become an excited electron, and the excited electron goes to the primary electron acceptor, and then it's passed along these protein carriers. So PQ, then it goes to the cytochrome complex, then it goes to PC, then it moves on to the chlorophyll A of photosystem 1. And chlorophyll A of photosystem 1 is called P700. Okay. So during this process, when the electron is passed along this electron transport chain, energy is going to be released in the form of ATP. And also when the electron reaches P700, chlorophyll A right here, it needs to be re-excited. The electron, it ran out of energy, so photosystem 1 is also going to accept a photon of light, which is passed along the light harvesting complex, and it re-excites the electron in P700. The excited electron goes up to the primary electron acceptor, and that excited electron is then again carried along by a protein carrier, eventually reaching the final destination, which is NADP+. So what happens when the excited electron combines with NADP+? Well, the electron is not the only thing that's combining with it. We have a hydrogen ion that combines with NADP+, and when all of these combine together, they're going to form NADPH. So NADP+ was reduced into NADPH by our enzyme right here called NADP+ reductase. Okay, so we kind of went over all the 8 steps in that diagram, but again, if we go through the specifics, so again in step 3, water is split by the enzymes and the electrons are transferred from the hydrogen atoms to P680+, thus reducing it to P680. And P680+, interestingly, is the strongest known biological oxidizing agent. The H+ are released into the thylakoid space. So what happens to the hydrogens after we split water? We split water into oxygen and hydrogen and also the electrons. So the hydrogens are going to be released into the thylakoid space, which is the empty space inside of the thylakoid, and the oxygen is going to be released as a byproduct. And as we saw earlier, the electron is going to eventually go to NADP+ and become NADPH. So number 4, each electron falls down an electron transport chain from the primary electron acceptor PS2 to PS1. Energy released by the fall drives the creation of a proton gradient across the thylakoid membrane. So that ATP that we saw earlier is going to be used to pump the hydrogen ions from the stroma into the thylakoid space. The potential energy is stored in the proton gradient, which drives the production of ATP by chemiosmosis. And just like cellular respiration, ATP is going to be produced by an ATP synthase enzyme in the thylakoid membrane. And that ATP synthase enzyme, remember, is going to combine ADP plus P in organic phosphate to make ATP. But in order to do this, we need energy plus energy, and that energy is going to come from the movement of the hydrogen ions down its concentration gradient, and that is known as chemiosmosis. In step 6, in PS1, like PS2, the transferred light energy excites P700, which loses an electron to the primary electron acceptor. P700 plus, P700 that is missing an electron accepts an electron passed down from PS2 via the electron transport chain. So each electron falls down an electron transport chain from the primary electron acceptor at PS1 to the protein ferredoxin, which is a protein carrier. And NADP+ reductase is going to speed up or catalyze the transfer of electrons to NADP+, reducing it to NADPH. The electrons of NADPH are available for the reactions of the Calvin cycle. This process also removes hydrogens from the stroma. Okay. So here's kind of an analogy as the electrons move from photosystem 2 to photosystem 1. So we can see how the electron is going to be excited by the particle of light called the photon. So it jumps from a ground state to an excited state. So we have an excited electron right here, but as it moves through the protein carriers in the electron transport chain, it's going to lose energy. So the electron has to be re-excited by a photon, and the electron can go back to an excited state, and that excited electron is going to eventually reach NADP+, And NADP plus also combines with a hydrogen ion to produce NADPH. And remember, when the electron is passed along the electron transport chain, we create ATP, and that ATP is used to pump hydrogen ions into the thylakoid space. Okay. So again, we see a simpler diagram of the movement of electrons along the photosystems, photosystem 2 to photosystem 1, and eventually to NADP+. So we need to split water into oxygen, hydrogen, and also an electron. So we have the electron here. And we need light or solar energy to give a photon, and the photon is going to be passed along eventually to the electron in chlorophyll A. Then we see the movement of the electron eventually all the way to NADP+. So the final electron acceptor in photosynthesis is going to be NADP+ versus cellular respiration where it was oxygen. So opposed to linear electron flow, in cyclic electron flow, the electrons cycle back from ferredoxin to the photosystem 1 reaction center via a plastocyanin molecule, abbreviated PC. The cyclic electron flow uses only photosystem 1 and produces ATP, but not NADPH, and no oxygen is released. Okay, so we can see how photosystem 2 is out of the picture. It's out of the picture. Okay. Some organisms, such as purple sulfur bacteria, have photosystem 1, but not photosystem 2. This is why they go only through cyclic electron flow. So it's thought to have evolved before linear electron flow, and cyclic electron flow may protect cells from light-induced damage. So here's a comparison of the chemiosmosis that happens in chloroplasts versus mitochondria. Chloroplasts and mitochondria generate ATP by chemiosmosis, the movement of the hydrogen ions down their concentration gradient after creating a hydrogen gradient, but they use different sources of energy. So for mitochondria, they transfer chemical energy from food to ATP. In chloroplasts, they transform the light energy into the chemical energy of ATP. Essentially, these reactions are reversed if you compare cellular respiration versus photosynthesis. The spatial or positional organization of chemiosmosis also differs between chloroplasts and mitochondria, but also there's a lot of similarities. So in mitochondria, we saw that the hydrogen ions, or the protons, are pumped into the intermembrane space. So they're kind of pumped to the outside, and they're pumped back into the matrix, the inside, through ATP synthase, driving the synthesis of ATP. In chloroplasts, this is the opposite. So the directions are the opposite. Protons or hydrogen ions are going to be pumped into the thylakoid space, and they're going to be pumped back out through the ATP synthase into the stroma, and that's what's going to drive ATP synthesis. So again, protons, that's referring to the hydrogen ions. So if we look at this diagram, we see how the movement of hydrogen ions or chemiosmosis is going to be different in different directions. So for the mitochondria, we can see how we have a higher hydrogen ion concentration in the intermembrane space. And for the chloroplast, it's going to be the thylakoid space. Intermembrane space is more outside, and the thylakoid space is going to be more inside. And we have a lower hydrogen ion concentration in the matrix, which is going to be more inside the mitochondria, and the stroma is going to be more out of the chloroplast. So, but the same thing is going to happen. We have the hydrogen ions. They're going to go through the electron transport chain, or they're going to be pumped along the electron transport chain, powered by the movement of the electrons into either the intermembrane space or the thylakoid space. And then they're going to be pumped back into or out to the stroma or into the matrix through ATP synthase. And that's when that movement of hydrogen ions back from the higher concentration to the lower concentration is called chemiosmosis. And that's going to drive the synthesis of ADP plus PI into ATP. So ATP and NADPH are produced on the side facing the stroma, where the Calvin cycle takes place. But remember that ATP and NADPH are both products of the light reactions. So these products of the light reactions are going to be fed into the next part of photosynthesis, which is the Calvin cycle. So light reactions part 1, Calvin cycle part 2. So in summary, light reactions generate ATP and increase the potential energy of electrons by moving them from the splitting of water into NADPH. The H pluses and the electrons being transferred to NADPH, and the oxygen being a byproduct. So let's move on to the second part of photosynthesis, the Calvin cycle. So we can see how the products ATP and NADPH from the light reactions, they're going to be fed into the Calvin cycle. And notice also that carbon dioxide is going to be another input into the Calvin cycle because the carbon from the carbon dioxide is going to provide the carbon for the glucose that we are going to make from the Calvin cycle. Okay. So here we see again a more zoomed out picture of the thylakoid membrane and the electron transport chain, along with the ATP synthase right here. So remember, this is the thylakoid membrane. And after we produce NADPH and ATP, these are going to be fed into the Calvin cycle, which is here located on the outside of the thylakoid, which is the stroma. So the Calvin cycle, like the citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. The Calvin cycle is anabolic, meaning it builds more complex molecules by using energy, and in this case ATP, in order to do so. And the reducing power of electrons carried by NADPH. Okay. Carbon enters the cycle as carbon dioxide, because carbon dioxide has carbon, and it's going to leave the Calvin cycle as a molecule called G3P, which stands for glyceraldehyde 3-phosphate. So why is G3P important? Because G3P is going to be used to make glucose by the plant. For net synthesis of 1 G3P, the cycle must take place 3 times, fixing 3 molecules of CO2. The Calvin cycle has 3 phases. The first phase is called carbon fixation, and this is catalyzed by an enzyme called rubisco. The second phase is reduction, And the third phase is regeneration of the CO2 acceptor, which is called Ruby P. So here's the Calvin cycle. So what are our inputs? Our inputs are CO2. And one CO2 is going to enter per cycle, but we need to go through 3 cycles. The first step is CO2 has to combine with another molecule called Ruby P, right here. And Ruby P, as we can see here, has 1, 2, 3, 4, 5 carbons. And Ruby P is going to combine with CO2 to form another intermediate of glucose, or sugar intermediate. So now we have 1, 2, 3, 4, 5, 6 carbons. So this process, this reaction is catalyzed by rubisco, which is an enzyme. Okay. So this 6-carbon intermediate is going to be converted into another intermediate called phosphoglycerate, and phosphoglycerate is going to be converted into 1,3-biphosphoglycerate. 1,3-biphosphoglycerate is converted into G3P. So the first phase, carbon fixation, that's going to be when carbon dioxide is going to combine with our Ruby P here. But phase 2 is when phosphoglycerate is going to produce eventually G3P by going through several reactions, particularly reduction. So ATP is going to be used to phosphorylate 3-phosphoglycerate into 1,3-biphosphoglycerate. NADPH is going to be broken down or oxidized into NADP plus and also hydrogen, and the hydrogen is going to be added onto 1,3-biphosphoglycerate, and that's called reduction. And so our final product in phase 2 is G3P. So why is G3P important? We see that G3P is important because eventually it's going to form glucose. But notice here, we actually are going to produce 6 of the G3P here. So 6 G3P. So only one of those G3P are going to be used per cycle for the formation of glucose. The rest of the G3P, as we see here, 5 of the G3P, is going to go to phase 3, which is called regeneration of Ruby P. So the 5 of those G3P are going to be converted into Ruby P, 3 Ruby P. And in order for us to convert it, we need ATP, the breakdown of ATP, and also the phosphate is going to be used to phosphorylate G3P in order to convert it to Ruby P. So the regeneration of Ruby P, the goal is to produce Ruby P once again, and the cycle can continue. And this is the Calvin cycle. Dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. So how do plants prevent dehydration? The evaporation of water, and as a result, the loss of water. Okay. Plants do this by closing their stomata. And as we saw earlier, stomata are those pores, which is the entryway or the exit way for oxygen or CO2, depending on which gas you see. So when the plants close their stomata on very hot and dry days, it conserves water by preventing evaporation. but because it closes the stomata, it actually cannot take in CO2, or it can't exhale oxygen. So because of this, there's going to be a buildup of oxygen, and there's going to be a lack of CO2. So because we can't intake CO2 and outtake oxygen, there's going to be a buildup of oxygen, and there's going to be a, let's say, absence or lack of CO2, which is needed for the Calvin cycle. And we can see that in the third bullet point: reduces access to CO2 and causes O2 buildup. So these conditions favor an apparently wasteful process called photorespiration. So when the stomata close, the plants are going to go through this photorespiration process instead, because it lacks the CO2 needed for the Calvin cycle. So far we've been going through the process of photosynthesis for C3 plants. So there's different categories of plants: C3 plants, C4 plants, and CAM plants. And the reason why they're called these different types of plants is because they go through a different type of photosynthesis. So C3 plants go through something called C3 photosynthesis. So this is most plants. Most plants are C3 plants, and they're called C3 plants because in the second phase, or I'm sorry, in the first phase of the Calvin cycle, the carbon dioxide fixation, when that's finished, we form a 3-carbon compound, the 3-phosphoglycerate. In photorespiration, when the stomata closes, Rubisco adds oxygen instead of CO2 in the Calvin cycle, producing a 2-carbon compound. Photorespiration consumes oxygen and organic fuel and releases CO2 without producing ATP or sugar. So that is why this is called a very wasteful process. Photorespiration may be a evolutionary relic because Rubisco first evolved at a time when the atmosphere had far less oxygen and more CO2. Photorespiration limits damaging products of light reactions that build up in the absence of the Calvin cycle. And in many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. So what's the difference between C3 and C4 plants? Okay. So in C4 plants, after carbon fixation, in the Calvin cycle, a 4-carbon compound is going to be formed instead. And in C4 plants, they minimize the cost of photorespiration by incorporating that CO2 and saving it in that 4-carbon compound. So there are 2 distinct types of cells in the leaves of C4 plants. So not only do we have the mesophyll cells, but we have another type of plant cell called the bundle sheath cells. So the bundle sheath cells are arranged in tightly packed sheaths around the veins of the leaf, and the veins are going to transport water. And the mesophyll cells, again, are the cells with most of the chloroplasts are loosely packed between the bundle sheath and the leaf surface. Sugar production in C4 plants occurs in a 3-step process. The production of the 4-carbon precursors is catalyzed by the enzyme PEP carboxylase in the mesophyll cells. So carbon dioxide fixation is going to occur in the mesophyll cells, and this is going to be helped by the enzyme PEP carboxylase, or catalyzed by that enzyme. So PEP carboxylase has a higher affinity for CO2 than Rubisco does. It can fix CO2 even when CO2 concentrations are low. And that is the reason why PEP carboxylase is used instead of Rubisco in the fixation of the 4-carbon compound. So these 4-carbon compounds are exported to bundle sheath cells. So notice how we went from the mesophyll cell to the bundle sheath cells now. There's a partitioning of different cells, 2 different locations. And within the bundle sheath cells, they release CO2 that is then used in the Calvin cycle. So basically, we were saving up that CO2 in that 4-carbon compound, and we release it when we need it again. In the situation where the stomata is closed and the plant can't breathe in CO2 through the stomata. So here's the anatomy of the leaf for a C4 plant. So you can see how kind of the mesophyll cells are around the bundle sheath cells. They're kind of in close proximity to each other. But most noticeably, we see that the bundle sheath cells are arranged around the vein of the leaf. To the right, we see the mesophyll cell, and we see the bundle sheath cell. There's a partitioning of locations here between 2 cells. So the mesophyll cell is going to be responsible for making our 4-carbon compound here, malate. So PEP carboxylase is first going to help fix CO2 into a 3-carbon intermediate and make oxaloacetate. Oxaloacetate is going to be converted into malate, and malate is going to be our intermediate that goes into the bundle sheath cell. So malate is going to be kind of a storage molecule for CO2. And now when we need CO2 in the bundle sheath cell, it's broken down into CO2 and pyruvate, which is 3 carbons. The CO2 can be then used in the Calvin cycle to eventually make our sugar. Okay, so you can kind of see how even though the stomata is closed and CO2 can't come in, we can store that CO2 in malate right here in the absence of CO2 and release it when the plant needs it to make sugar. Since the Industrial Revolution in the 1800s, CO2 levels have risen greatly. Increasing levels of CO2 may affect C3 and C4 plants differently, perhaps changing the relative abundance of these species. The effects of such changes are unpredictable and a cause for concern. So suitable agricultural land is decreasing due to the effects of climate change, while the world demand for food continues to increase. C4 photosynthesis uses less water and resources than C3 photosynthesis. So scientists have genetically modified rice, a C3 plant, to carry out C4 photosynthesis to increase its survivability. They estimate 30 to 50% increase in yield compared to C3 rice. So that's actually pretty cool. So scientists have converted rice into C4 plants, and this increases the yield or amount of rice that's produced. Finally, we have the third type of plant. So some plants, including succulents, use CAM metabolism to fix carbon. So CAM plants open their stomata at night, incorporating CO2 into organic acids that are stored in the vacuoles. And during the day, when it's hotter, the CO2 is released from organic acids and used in the Calvin cycle. Okay. So why is the stomata closed at night? Why is the stomata open at night? Because at nighttime, it's cooler. So less water is going to evaporate. So water is going to not evaporate. But during the day, the stomata is going to close. And that's because during the day it's very hot and more water is going to evaporate. So that leads to more water loss. Okay. So the CAM pathway is similar to the C4 pathway in that we're trying to store that CO2 into a 4-carbon compound. And we release it when we need it. We release the CO2 and use it in the Calvin cycle. So the C4 pathway structurally separates the initial steps of carbon fixation from the Calvin cycle. So remember, in the C4 pathway, we had 2 types of cells. And we had because of that, we had 2 locations. But in the CAM pathway, these steps occur in the same cell. So we only have 1 cell type. And the time is going to determine when CO2 is going to be released. Okay, so in this photo we have a C4 plant versus a CAM plant. We can see how the C4 plant has 2 different types of cells, and photosynthesis is going to be partitioned between these 2 cells. So this is based on the location. However, for the CAM plant, photosynthesis is going to be partitioned based on time. So CO2 is going to be fixed onto our intermediate during the night. So we're going to have a 4-carbon compound. And during the day, when the stomata is closed, that 4-carbon compound is going to break down and release CO2 where it's needed for the Calvin cycle to produce our sugar. So during the day, this is when the stomata is closed, and we cannot obtain that CO2 from the environment. So again, for the C4 plant, remember, this is not dependent on the night or the day. Photosynthesis is going to be partitioned based on the location of the cell. And so finally, this is just a review of what we went over. Why is photosynthesis important? Well, life depends on photosynthesis. And firstly, the energy entering chloroplasts as sunlight gets stored as chemical energy in organic compounds. So we remember that the chemical energy is going to be stored in the form of glucose. And the sugar made in the chloroplasts supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. So that glucose is eventually converted into ATP, which is another form of chemical energy, and ATP is going to be used by the cell to synthesize other molecules. And plants store excess sugar as starch in chloroplasts and other structures such as roots, tubers, seeds, and fruits. So remember that extra glucose, if it's unused, it's going to be stored as a polysaccharide, which is starch.